Hello, everyone. I'm sorry, I'm a bit sick. My voice is a bit different today. Today, I'm going to talk to you about BBB permeability measured with ASL. So I'm going to give a bit of introduction why blood-brain barrier permeability is important in Alzheimer's, how can we measure it with ASL, and then show our results regarding cognition and amyloid status. I work with Inkian and Ian Petter and Frederick Parkhoff in Amsterdam. Okay, so we know that Alzheimer's disease is characterized by amyloid, tau, and neurodegeneration. And especially recently, there was this NIA criteria, the announcement of the new criteria in July in AIC in Amsterdam, where they also acknowledge that vascular damage and inflammatory processes also make part of the Alzheimer pathophysiology and can be used to diagnose Alzheimer's earlier. And also, we all know that the glymphatic system is very important for the Alzheimer's pathophysiology, because we know that it's responsible for the brain clearance, and we know that amyloid accumulates within the neurons, so brain clearance is very important for the removal of amyloid from the brain. And all these three concepts have something in common, which is the blood-brain barrier. We know that the BBB is this endothelial layer of cells that surrounds the vessel of the brain and protects the brain against toxins in the circulating blood, and is responsible for inflammatory processes and the glymphatic system. And we want to investigate its integrity in early stages of Alzheimer's. So we want to use some information that BBB permeability can give us and try to use it as an early biomarker of Alzheimer's. One of the hypotheses is that the BBB will trigger neuroinflammation that will promote accumulation of amyloid beta, and also together with leakage of the circulating toxins from the blood plasma into the CNS, it can cause neural damage and then give rise to the cognitive impairment and onset of dementia. And especially in the recent years, from 2021 on, this concept of brain clearance and glymphatic system has been investigated as well as a very important part of the whole process. But then how can we actually measure the BBB permeability? The current biomarkers include some of the ones that I'm going to explain now. So we have post-mortem investigations on BBB that we actually know, okay, BBB is disrupted in Alzheimer's disease, but of course, it only gives us information of the end stage. So we cannot do anything with it. So in vivo, we can use albumin ratio that is widely used for BBB disruption of permeability, because albumin is a protein that must be in the blood. So if it crosses to the BBB and it is increased in the CSF, this ratio will increase. But of course, it's one value, right, for the whole brain. So it does not gives us information about the localization of the leakage. For regional analysis, we can use CT and PET, but of course, we know that they use contrast agents or radioactive tracers. So MRI becomes a useful method for these studies. So first of all, I'm just going to briefly talk about how can we measure BBB with gadolinium. So you probably know DSC and DCE. So depending on the extent of the BBB leakage, the gadolinium will cross and it will accumulate in the extravascular space. And since we know that it will change the T1 relaxation times of the tissue, basically with a pre and a post contrast T1, we can see exactly where the leakage is, right? And this is widely used for tumors, because when you have a tumor, the blood-brain barrier is all disruption, opened holes everywhere. So the gadolinium can easily cross, and then we can very clearly see where it is. However, we know that also gadolinium is invasive, and it has been shown to accumulate in the body and possibly in the brain, and this has some consequences. So we want to do something that is non-invasive and something that can explain us more about initial stages, because gadolinium is a larger molecule, so it will only cross in later stages of BBB disruption, right? So what if we use water? The idea is to use water as a tracer here, as a contrast agent in a way. And so we want to do this with ASL MRI. You probably know a little bit about ASL. So we label the water molecules in the feeding arteries of the brain. And after some time, called the post-labeling delay, we acquire an image when the water is reaching the brain parenchyma, right? So, and then we can have and quantify the cerebral blood flow. And this is a very nice image, very nice image. So normally you don't have this as good as we have here. And sometimes depending on if a person is older, of course, if you use the same PLD as a healthy adult compared to an elderly person, sometimes you'll have some macrovascular signal. So there are a lot of issues that we need to deal with. So I'm going to talk about that a little bit more. So, okay, so we know where the water is going, but how much water actually crosses? So this is what we want to measure. So we can use something that is multi-echo ASL, because we know that the water molecules in the blood and the water molecules that have crossed to the gray matter, to the brain tissue, they will have different T2 relaxation times. So if we use multi-echo ASL, and knowing that the T2 of the blood is longer, higher than the T2 of the tissue, we can basically have these two compartments where we can see, okay, these molecules are decaying like this, these molecules are decaying like this, so this amount of water crossed, okay? And so this is what we do. We use multi-PLD and multi-echo times ASL, and then we can calculate these BBB permeability maps. And this time of exchange is kind of the time that the blood takes to cross. So just a reminder that will stay there, I think, during the results. Like if the time is lower, so the time that the water molecules took to cross is lower, it means that there's higher BBB water permeability. But this is a bit vague, so I'm going to explain a bit more how we do all of this. We use time-encoded ASL instead of multi-PLD, normal multi-PLD. So the PLD is the time that we wait. So if you have very short PLDs, you still have the main, the labeled waters will still be in the main bigger arteries, so not in the capillary site yet. And if we wait too long, of course, with the T2, with the T1 relaxation, we won't have any more signal, right? So we need to kind of adapt this PLD, and this time that it takes, called arterial transit time, changes per person. So multi-PLD approach has been used. So what we do is we do control and label images, and we wait different times, and we acquire images after different PLDs. But we can also use time-encoded ASL that has a matrix of labeling and controls. This is a bit complex, but I have a slide at the end if you want to know how we do this properly. And then we do the readouts after different times. And basically, when we decode these images, so with different addition of or subtractions of these red images, we'll have the different PLDs. So it will give us the same information, but with much faster. And this is important. Why is this important? Because we want to add it to clinical protocols, and we know that doctors don't have time, so we want to make it as efficient and fast as possible. So we have this Hadamard 8 sequence, and we have a Hadamard 4 sequence, so 8 by 8 and 4 by 4. And the first one is better to, since we have more PLDs, we can better quantify the CBF. And then with Hadamard 4, we have multi-echo times to then create and calculate the time of exchange through the blood-brain barrier. So the multi-PLD is to have a better CBF, and multi-echo for the BBB permeability. Okay, so this sequence was created by Matthias Gunther in Germany, and they consider that per voxel we actually have 3 compartments. We have one compartment, so the signal in each voxel might come from the arteries still, or already in the capillary sites, but it can be intravascular or extravascular. So we have 3 equations here. The first one is for the first part, the second one is for the intravascular space, and then the third one I'm going to explain a bit better. We have the signal that is coming from the extravascular space, which is what we actually are here to measure. This magnetization relaxation function of blood and tissue, we assume the T2. So this is not subject specific. This is one of the limitations. We are trying to improve on this because we assume that the T2 of blood is 165 milliseconds and that the T2 of tissue is 85 milliseconds. And then from this third equation, we have this ratio there that will give the ratios like how much is actually going from one side to the other. And then we have the time of exchange, which is the value that we want. So we assume we use the perfusion, the M0, the arterial input function, and these magnetization relaxation functions that for which we use the T2s that are the standard values, okay? And then we take the time of exchange and we put everything into this model from FSL. And here you can see also that they consider, so first the signal enters the arteries and then it still takes a bit of time. This is the ITT until it goes to intravascular and then extravascular. Okay. So with this, we have per voxel one value of time of exchange. And then we can create a map of the whole brain time of exchange. And this time, again, if the time is lower, means more BB permeability. Okay, so this is the background, the explanation of the sequence. Now our project, the DEBI project, wants to use this sequence and add it to MRI protocols to use it as a non-invasive early biomarker of Alzheimer's. We are doing a lot in reproducibility, accuracy, and normal variability. And now I'm going to show you some results from this clinical question. So how do the values change between healthy controls and MCI patients, which is mild cognitive impairment, so not already Alzheimer's disease, but they already have some cognitive impairments. And also the 2B is AD biomarkers. In this case, we have amyloid. Okay, so for this, we used two cohorts acquired in the same scanner. So scanning the same scanner. So we have 77 cognitively normal from the LCBC and 39 from DDI. And from this 39, we have 24 cognitively normal and 15 MCI. ASL and T1, Hadamard, pseudo-continuous 3D Grace readout, some specifications of the sequence there. And then we use the CBF and the TEX maps. And then we also have the amyloid status. So some of them had PET, some of them had CSF, and based on specialized readers, they said, okay, this patient is amyloid positive or amyloid negative. And so our idea was then to understand if amyloid status was predictive of CBF and TEX, corrected for age and sex, and cognitive staging that in this case was controls or MCI. Okay, so some demographics. Both groups have around 64, 67 years old. This is like all the values of time of exchange over age plotted together, and we can see that there's a decrease of time of exchange over age. So again, less time, more water permeability. I don't know why it's not here. And then also CBF. Here CBF is not exactly significantly decreasing over age, but we also see some very high and very low values here. So we still need to check all of this. And as expected as well, the CBF also is higher for females than for males, and time of exchange as well. So that's why we also corrected for age and sex in all the analysis. So this is just a mean of the time of exchange maps from people that were healthy controls and amyloid negative. So we see that the values are a bit higher than when we have MCI, the subjects that were MCI and the subjects that were amyloid positive. And these are the correspondent CBF maps. We also see that it makes also sense that the NC and amyloid negative had higher CBF, but these maps don't look very good also because we are using all the PLDs and we want to kind of QC these images and create CBF maps from the most important PLDs per subject as well to have a more reliable CBF maps. Now to the results. So we have two groups, amyloid negative and amyloid positive, and we did time of exchange of the whole gray matter, 1 value per person, and this is what we see. So this is gray matter text, amyloid positive are lower values, and then we also see that there's a lot of difference between males and females. And after performing linear regression, we basically see that even corrected for age and sex, the amyloid status is still decreased in amyloid positive subjects. And for CBF, we actually see an increase in amyloid positive, even corrected for age and sex. What we think that is happening here is that amyloid positive creates some kind of inflammation reaction, and then CBF increases in cognitively normal people. We have a previous work where we also saw this, and there are other papers where they see, okay, this person is cognitively normal, is amyloid positive, so actually the CBF is increasing a little bit to compensate for this. Then also the text corrected for CBF, because we know that there's some kind of dependency between text and CBF. So even corrected for CBF, text is still predicted by amyloid positive, which is very interesting. And CBF as well, corrected for text. Okay, then the cognitive staging. Here, so all of the LCBC participants were cognitively normal, and we separated them from the cognitively normal of the DDI, which were the other group, because they were also younger. And what we see is that actually text decreases with more, so mild cognitive impairments is this group where you have some neurodegeneration, but it's not dementia. And so again, corrected for sex and age, we see that the MCI group is different from the controls. For CBF, it's a bit more messy, so not very clear what is happening. But I just wanted to show you. So for those who are not used to these R outputs, this is basically the p-values are here. This is the beta, so the estimate. And so and each one of these are the ones just, so it always uses one group as reference, and in this case, the LCBC normal controls is the reference group, and then you have the MC and MCI from DDI. And so even corrected for age and sex, there's always some difference. Okay, so you know that the glymphatic system, so this is one of the images of one paper that explains how the glymphatic system works. So we have the capillaries, and then these are not the capillaries yet, actually, because I was discussing with Hang Zhang Lu yesterday, and capillaries don't have perivascular spaces, and here we do have perivascular space. But all this movement of the water getting out of the blood to the tissue happens even before the capillary site, and then the toxins are removed to the venous parts. What we see is that since TEX is lower when permeability is higher, what we see is that actually with amyloid positive, the BBB is passing more water. So this might be explained by, you know, the brain wants to clear the amyloid that is being accumulated, so the water needs to cross more. I don't know if I have another conclusion. Yeah, so this makes sense with our theory that lower time of exchange means higher BBB water permeability, which makes sense, so less time, more water permeability. But there's another group where they use diffusion-weighted ASL instead of multi-echo ASL. So they use B values to kind of separate the two compartments. And they see a bit of the opposite of what we see. And their explanation is that there's actually less water crossing with disease because the aquaporin channels, which are the responsible for the water permeability, are dysfunctional. So we are still trying to understand why they have some results and we have others. But this was just to show you that we are working on it. And if you have some feedback or questions, please let me know. And thank you for your attention.